Hello, everyone. Welcome to this first keynote of e-Rum 2020. So I'm really happy to have on stage today our first keynote speaker, and she is Stephanie Hicks. Stephanie Hicks, she is an assistant professor at the Department of Biostatistics at Johns Hopkins Bloomberg School of Public Health. Welcome, Stephanie.
Well, hi, thank you. So excited to be here.
That's great to have you here.
First of all, just making sure.
Yes,
okay.
I was just presenting you, and I mean, saying that you're an assistant professor at Johns Hopkins and a faculty member also at Johns Hopkins Data Science Lab, also a very active in the social stream of our co-host of the Corresponding Author podcast, co-founder of Our Ladies Baltimore, and also very active in the Bioconductor community, if I'm not wrong.
That is all correct, yes.
Great, that's great to have you here. And she's here today. Today's talk, Stephanie keynotes, is about something very important, using R and data science to improve human health. So I'm really eager to see and to listen to your words. Just a little reminder to our audience that we can also use this system, Slido. So join at slido.com with the code e-Rum2020 and ask questions. You can ask questions there while the talk is going on, and also upvote others people's questions. So we will have a Q&A session just right after the talk. Okay, so thank you, Stephanie, for being here, and I leave you the floor for your talk.
Okay, see if I remember how to do this. And okay, are you looking at my slides?
Yes.
Okay, and let me turn off my camera. All right, does everything seem okay?
Everything is good.
Okay, perfect. All right, well, hello everybody and welcome. I'm really excited to be here. I just wanted to say thank you so much to the program committee for the opportunity to present on how I use R in the life sciences. So I just will mention that my slides are available at the link below. Okay, so when I received the invitation to come give a talk today, one of the things I immediately thought about was the Milan Cathedral and being able to visit it. I've read about it in books and I've read about it online, and I was really, really excited to come see it in person. And the world that we live in did not let that happen today. So we live in a world, as we all know, as we heard about this morning from the COVID art contest winners and awardees, that coronavirus hit. And in particular, Italy, for example, was one of the hardest hit regions or countries outside of China in February and March, with over 600 cases in February, rising up to 15,000 in March. But with this tragedy came a new opportunity, for example, as I mentioned, to host this COVID R contest, where this contest was designed to develop open source R products like Shiny apps to explore data around the pandemic. I actually watched the whole event this weekend as I was preparing for this keynote, and I really enjoyed seeing all 15 of the presentations. And I just wanted to say congratulations to the winners and the awardees at the conference. Well done, well done. But as I was watching this, one thing that popped in my mind is I'm curious, like, what is the motivation that these developers and other developers around the world for creating these R resources around the pandemic? And so, as any good researcher does, I turned to Twitter over the weekend to ask, for example, for those who've downloaded, explored, or analyzed any COVID-19 data, looking at plots, web apps, dashboards, papers, what was your motivation? So a couple of ideas that popped in my head was maybe an individual wanted to help make an informed decision for an individual, a business, a city, or a country. Or maybe you wanted to just help a loved one, or maybe help humanity, or maybe it's just pure curiosity. And so this was interesting poll response. A lot of people seem to be just really curious. And then there were some comments that were left, such as maybe for academic research, a hackathon or competition job for government agencies, such as Ministry of Health, programming, visualization portfolio, or an exercise for a data science course. That made a lot of sense to me. Another one that I read was, I gathered some references, methods, and tools to assess when the local healthcare system would collapse. And when I read that, it got me thinking about healthcare systems and in general, and how we as the world are trying to overcome this pandemic. So I think one of the biggest ways that we are trying to overcome this pandemic, in addition to like social distancing, is to create vaccines, to produce an immune response in our own bodies whenever we encounter the coronavirus. To date, there are over 150 vaccines of SARS-CoV in various stages, with the first trial starting, I think, in March 2020. Now, as we all know, it typically takes years to research and test vaccines before they reach the clinic, but there are some scientists who are trying very hard to reach a safe and effective vaccine by early 2020. So I thought this was a great motivation to be able to introduce a little bit of biology. And so to do that, I thought, well, let's talk a little bit more about vaccines. How do you even create a vaccine? Well, it turns out there are many different ways to create a vaccine. For example, you could have a genetic vaccine, and this is a vaccine that where you use one or more of the own coronavirus's genes to provoke an immune response in our body. So the virus is introduced into our bodies, and the goal is for our body to produce an immune response to attack it. As opposed to a genetic vaccine, you could have a protein-based vaccine. So here you're using a coronavirus protein or a protein fragment to promote an immune response. You could also use a viral vector. So here you're using a virus to deliver the coronavirus genes into the cells. Or you could actually use the whole virus itself, just a weakened or an inactivated version of it. Now, as clinical phase 2 trials start to emerge, what will happen is we may start to see how different subportions of the population, for example, children versus elderly, might respond differently to vaccines. And you might ask, well, why is that? Well, to do that, we need to talk a little bit about biology. So I'm going to use this COVID-19 example as a motivation to talk about biology at a whole, and I'm going to switch from the concept about COVID vaccines to a broader question about what makes us diverse as individuals in a human population, because this is essentially what will drive how we end up responding differently to these vaccines. So if you recall, in my high school biology class or your high school biology class, for example, this story, to answer this question about what makes us diverse, this story starts with what's known as the central dogma of biology. So at the top here, we have a piece of DNA. Can you see my mouse by chance? I can't tell. I'm going to assume no. So we have a single piece of DNA here at the top. This DNA, or portions of that DNA, get converted into what's called RNA. It's also, you can think about it as a gene. That process goes through something called transcription. And then an RNA gets converted into something called proteins via translation. And so I've mentioned a little bit about these words when I was referring to the different types of vaccines. So whether you're using a coronavirus protein or a coronavirus gene, such as it in the DNA or in the RNA. Now, genetics is interested in looking at changes in the DNA and, of course, associating it with some phenotype. So they're interested in identifying if there's a small change in a base of my DNA, how does that change in the phenotype that I observe? So I'm going to pick on my colleague, Jeff Leake, for just a minute. Here are two pictures of species with different genomes, and they have two different phenotypes. And if you know anything about Jeff Leake, you know he's the exact opposite of a sloth, so this is a good analogy. Now, if we go back to the central dogma of biology, genomics is interested in how all of these things interact. For example, we're interested in looking at changes in the RNA or changes in the DNA, or how protein interacts with the DNA. So protein can go sit back on the DNA and cause it, for example, to go through additional transcription to create more RNA, which will then create more protein and so forth. And of course, associating all of these changes with some kind of phenotype. So let's talk about how genomic data is generated. So to answer that question, I'm going to start back at a piece of DNA. DNA, what we do with it is we fragment it up. We tear it up into tiny little pieces, and then it goes through an experimental protocol to produce what are called sequencing reads or reads. And they are essentially little short fragments of 50 base pairs or 100 base pairs or 200 base pairs or even longer that we use as the output from our sequencing experiment. So this is the output that we start with, these raw reads from the sequencing experiment. Now, turns out each biological experiment can have files with millions or billions of reads. And you could ask, what do we do with these reads? Well, the first thing we have to do is we have to figure out where these reads come from. So if we have some kind of reference genome, like a human healthy reference genome, we could use that to figure out where these reads are coming from. So that's exactly what we do. We start with a piece of reference DNA, and on it sits a gene, for example, so gene XYZ. What we get out of a sequencing experiment are a set of reads. And the goal is to try and figure out where these reads come from. So what happens is we compare the similarity of the A's, the T's, the C's, and the G's from the reads to the A's, the T's, the C's, the G's from the reference genome. And then what we do is, once we're confident about where they come from, we essentially count them. We say how many reads are coming from gene XYZ. And in this case, if I did my math correctly, there are 23 reads coming from gene XYZ in this one sample. So what do we do with that number? We put it in a matrix. So we start by recording these 23 reads that are coming from gene 1 in sample 1. And then we can repeat this for counting the number of reads that come from gene 1 and more samples, however many samples we have. And here it's 6 samples. Then we can repeat this process and we can count the number of reads that come from gene 2 in sample 1, 2, 3 through 6. And we can repeat this process up until we have gene G, which is usually around 20,000. We have around 20,000, for example, protein coding genes in the human body. So this is the matrix that most people are maybe familiar with if you've ever worked with genomic data. But I just want to point out there's also another piece of information. You can think about this as metadata. That's also really important. It's essentially very important bookkeeping. It's information about where the gene comes from. So does the gene 1 come from chromosome 1? And what is the position that the gene starts? And what is the position that the gene ends on? I'm going to talk more about this metadata in a few minutes and how Bioconductor handles it. But in general, this is the type of data that we analyze. And this data can be data that's stored in a CSV file, or it could be data that's stored in a database somewhere and we're bringing it in, or streaming it in, or it could be data that are stored completely on disk, such as an HDFI file. It doesn't really matter. This is just a matrix of counts that we're typically working with. And we can ask interesting questions like, what is the average expression of gene 1 in samples 1, 2, 3, and compare that to the average expression in gene 1 in samples 4, 5, and 6. And a way we could ask for if there's a difference in the average expression between these two groups is something like a t-test, for example. All right, so how can we actually analyze this genomic data? I've now given you a sense of what are genomic data and how do we, what might we be interested in doing with it? Well, the way that I work with it is Bioconductor. So if you've never heard of Bioconductor, you can think of Bioconductor kind of like as a cousin to the CRAN project, but specific for the analysis of biological data. So formally, Bioconductor is an open source and open development software project for computational biology. Open source meaning anybody can read and modify the underlying code. Open development meaning anybody can contribute to the project. This began in 2001 by Robert Gentleman and others, and now Bioconductor actually includes 12 full-time core developers to build and maintain the core infrastructure. I think it has over 1,900 packages to date, so it's quite a big task to try and coordinate. But some big priorities for the project. They're really focused around reproducible research, addressing domain-specific challenges with high-quality documentation. So some examples. For example, each software package must contain a vignette. There is a huge community support forum that you can ask questions on problems and get answered by experts in the field. Bioconductor has a Slack workspace that's open to anyone. Many workflows to help users get started in analyzing biological data. Lots and lots of great teaching resources. And something that I really like is that Bioconductor strongly encourages the use of existing data and software infrastructure from Bioconductor packages to really enable interoperability. So there are a lot of great things about Bioconductor. And I think the mental image that I want anybody watching this to walk away with is that Bioconductor aims, I guess the mental image that Bioconductor is aiming for is that there are a lot of tools and packages for the analysis of genomic data, similar to the way there are a lot of individual musicians in an orchestra. And to make them all work together, Bioconductor functions as a conductor of the orchestra. It provides a flexible platform to integrate everything together. All right, so let's talk about a little bit what's in Bioconductor. So to do that, I thought I would showcase or highlight a package that I discovered in the last year or two called the Bioconductor or BioC package tools. And here I'm showing you how in one line of code, you can get a tibble or a tidy data frame of all the download statistics for Bioconductor packages. So for example, in row 1, you can get the number of downloads for the package AB array from January 2020, and there were 117 downloads. I will mention there's a column called software here, or the repo here with with software in it. Software is the biggest type of package that's contributed to Bioconductor, but there are actually different types of packages. So we could figure that out really easily. And because this is a tibble or a tidy data frame, you can leverage the tidyverse essentially to explore it. So I can take the package's tidy data frame and I can filter it for all the packages from 2020. And I can essentially group by repo and count. So you can see that, yes, software is by far the largest set of packages in Bioconductor, but there are also these things called annotation data or experiment data. Annotation data really streamlines this tedious bookkeeping that I mentioned earlier. So depending on what reference genome you're using, the location of a gene might change. And it's really important to have infrastructure to make that as easy as possible. And Bioconductor has really done that well with annotation packages. Experiment data packages, they contain process data, and they're also really useful for teaching. So if you want to demonstrate the use of a package or use of a function in your package, you can use experiment data to do so. So Bioconductor software packages have really been steadily increasing over time, all the way up through 2020. It's really quite nice. And yeah, I think there's around 1,900 or 2,000 packages to date. So what is this standard, or like what are the different types of standard data infrastructure in Bioconductor? So I think the one to start with is a genomic ranges or a G ranges object. And here I'm representing a chromosome, chromosome 1, and I'm showing you two different genomic regions from this chromosome. So on the left side, there are 4 columns, and on the right side of this object, there are 2 columns. On the left side, these are required by a G ranges object or genomic ranges object. And on the left side, you can put kind of whatever you want there. That's it's however you want to, whatever information you wanted to keep track of about each genomic region. But on the left side, you have the seq name column, which contains information about what chromosome the genomic region comes from, the start and the end position. I'm going to ignore the strand information. That's a little bit too nuanced for this talk. And then I've incorporated a gene ID or gene name and some score. And you can think about this score, for example, as the number of reads that map to a particular genomic region. It can be really anything. So to create a G ranges object, it's actually really easy to do. There's a package called Genomic Ranges, and in a few lines of code, you can have an object that Bioconductor expects to use in almost all of its packages. So it's really quite fun. So here I've got 2 genomic ranges similar to what I was showing you before. Things you can do with it, you can ask, for example, the width of the regions, or you could use base R syntax to filter rows and ask for rows that have a score greater than 15. I will give a shout out to the Ply Ranges package from Stuart Lee, Di Cook, and Michael Lawrence. They've developed a really nice API to be able to make genomic data analysis more human readable by being able to integrate the pipe from the McGritter package and really extending the dplyr package with action verbs such as filter and subset or select and things like that. So shout out to them for being able to make it more human, or genomic data analysis more human readable, but I'm going to skip over that for now. Okay, so let's talk a little bit about one type of genomic data that I work with, and this is gene expression data, or RNA. So the traditional way that people have measured gene expression is something called bulk RNA sequencing. And so for a given gene, you can see what you get is the average expression across all the cells in a tissue. So if I take a piece of tissue from the brain, for example, I can get on average how much RNA was expressed from a particular gene given all the cells in that tissue. You don't actually get to know what are the different cells that are present. An analogy that I like to use is Legos. So it's like you have a big pile of Legos, and for a given feature, such as the width of a Lego, you get to see an average of the widths across all the Legos in a pile. Now, turns out there are newer technologies, such as something called single-cell gene expression or single-cell RNA sequencing, and it's an advancement in the field because now we can capture all 20,000 features or genes in an individual cell. So we get information, new information about what genes are being expressed in each cell as opposed to an average across all the cells in a tissue. And it's really, really exciting because you can isolate these cells and you can use the cell profiles to do things like unsupervised clustering and group different cell types together and figure out if there are any novel cell types. An analogy that you could think about is with Legos is being able to separate the Legos and group them by size, color, and type. So using unsupervised methods, unsupervised clustering methods, for example, to figure out what are the different cell types in my big pile of Legos. So just a little bit more formally, single-cell RNA sequencing starts with a tissue, such as the tumor or brain tumor that I was referring to. You isolate and sequence the individual cells. What comes out of the experiment are these reads, and these reads are mapped to a particular position in the genome. So say gene 1 in cell 1. For a given cell, I can count all the reads that map to gene 1. And then from there, I record this in a matrix. So along the rows of this read counts matrix, I have gene 1, gene 2, gene 3, and on the columns, I have cell 1, cell 2, and cell 3. And then from there, we can do a lot of great things with this count matrix. We can, for example, compare the expression profiles of single cells, either using a heat map or some kind of dimensionality reduction, such as principal components. And there we hope to discover new cell types or figure out if there's more of a continuum of cell types. And a common question you might ask with this data is, for example, can we identify changes in genes that are expressed that are associated with differences between healthy and cancer cells? And so to do that, if you might ask, what does a healthy cell even look like? How do I know what I expect a healthy liver cell to be? Well, or how do cell types change across tissues? Or how does a cell change over time, for example, going through embryonic development? Well, there's an organization called the Human Cell Atlas, and they have a mission to create a comprehensive reference map of all human cells as a basis for both understanding human health and diagnosing, monitoring, and treatment of disease. It's a very large worldwide organization that's producing essentially a really, really big reference map of all cells in the human body. I'm not going to go into too many details, but this is essentially one way that we are going to be able to, for example, if I'm an oncologist treating a cancer patient, if I want to do single-cell RNA sequencing on a tumor from my cancer patient, I can compare the cells from the tumor to some kind of reference map. What do I expect brain cells from a healthy individual to look like compared to what the cells come from that I have, and trying to figure out what the differences are. So this is really exciting. But one thing I just want to note is that they wrote a white paper in 2017, and in it says they expect over 10 billion observations or cells by 2020 as kind of the starting point of this reference map. And what that means is that's a very big data set to try and process as a statistician or a data scientist. And so the challenge, one of the challenges that I wanted to just highlight from my work is being able to scale up to millions or billions of cells. So being able to write methods and software that can do that. Now, when you start to think about that, immediately there might be this trade-off that shows up in your mind. There's this trade-off between speed and accuracy. If I want to make things more scalable in a reasonable amount of time, then I'm probably going to have to lose some accuracy doing so. And I just want to note that methods for single cell data and in general that don't scale are going to probably not become, or not likely to be used as data become bigger, because they're just not going to be reasonable to run in an amount of a small amount of time. And I think it's also important to write software and methods that can work with any backend or any backend data format. So whether this is data on disk, or it's such as something like an HDF5 file where you never load the data into memory, or data in memory, or just sparse data, or data coming from databases and stuff like that. So this is that a 3-minute warning? Okay, I'm gonna assume so. Yes, okay. So this is a really big problem. For example, I pulled 4 papers from 2019. They all start with 1 million or more plus cells. One way that you can try and perform, for example, unsupervised clustering of single-cell RNA-seq data, or just in general of observations, is k-means. And k-means turns out to be not that scalable. So there was an algorithm introduced by Scully in 2010 called mini-batch k-means. And the idea is at each iteration, you use just a random subset of the data. And this is what I have been working on, or one project that I've been working on to make data scalable. So we wrote an R package along with my collaborators called mini-batch k-means, and I just want to highlight the coolness of it. So compared to k-means in red, comparing to mini-batch k-means with data stored on disk in blue, we are far more memory efficient as we scale the number of cells from 75,000 to 1 million, with the y-axis being the memory usage on the left and the time on the right. And so we're also not only saving memory, but we're also being able to cluster cells in a much faster amount of time. And while there's no reason, and there's no real reason why this couldn't be used more broadly outside of single cell, basically anything that you would use for k-means, you can use for mini-batch k-means, but our motivation was for single cell. And then the last challenge I want to talk about is education. So how do you analyze single cell RNA-seq data? So I frequently hear this when somebody asks me for the first time, I have new single cell RNA-seq data and I don't know really what to do. I wrote a paper to essentially address that. So I wanted to write this paper to help users and researchers navigate the world of single cell Bioconductor ecosystem of packages. It's a very rich system. I'll just leave it there in case you want to check it out. But we cover, for example, standardized ways to store single cell data. For example, the single cell experiment object is an S4 object that can have any matrix like object as a backend. So whether it's HDF5 files or data stored in memory or sparse data, it's really cool. We talk about Bioconductor workflows. But I think the most exciting part of this project was this online companion book that has really taken on a complete life of its own. It's written in book down. It's compiled on a regular basis to make sure all the code works. It has an enormous amount of philosophical discussions, mostly by Aaron Lunn, a gentleman by Aaron Lunn, about single cell analysis. It's truly cool. Most importantly, lots of R code to help you analyze your single cell RNA-seq data. And this is my last slide. I just want to leave you with a sense of how all this might be useful and where genomics is going. So as I mentioned, there's been a transition in the world of genomics from going from bulk to single cell RNA sequencing. But there are now recent advances that can capture gene expression in a spatial or 2D plane. And this is really useful because you can now identify where the tumor cells are and see how they're communicating with, say, immune cells that are sitting right next to it, because you know the 2D space of where these cells are sitting. But soon we're going to be able to reconstruct the original organ itself in a 3D plane, and maybe in a 4D plane as well when you include time. And I think the combination of advances with these experimental technologies, going from bulk to single cell to spatial to 4D, for example, but the combination with advances in scalable, accurate, reproducible methods and software using R and Bioconductor makes this a really exciting time to be working in. And I think this is a very important step as we go towards human health. And I will leave it there. Thank you, everyone.
Thank you, Stephanie. Thank you, Stephanie. You can be back in video too.
Okay, should I stop sharing my screen?
Okay, yes. Okay, thank you. Thank you for, I really loved the Lego analogy. I began recently to work with single cell RNA-seq data. I'm not an expert actually, but I was an expert in Lego, so I definitely have to switch to that.
I'm so happy to hear that.
I like it. So I'm going to share my screen with the Slido questions that in a moment here. Okay, so we have a winner. We have a few minutes, maybe something like 10 minutes for questions from the public. I would start with the most popular here. Can you comment on the integration between Bioconductor and Tidyverse? So that's a topic. Are there efforts to align these two frameworks?
Yes, there are actually. So I blew through it and I took out the slides for my presentation. For example, there's the Ply Ranges package by Stuart Lee, Dai Cook, and Michael Lawrence. And the whole point is to be able to make genomic data analysis more human readable by basically borrowing all the wonderful ideas from dplyr and the Tidyverse, but just bringing it in for objects that Bioconductor is used to. So that's one way that it's happening. I would say that if you are, so in my own day-to-day work, I'm often exploring data using the Tidyverse itself. For example, being able to make plots really easily. However, there's a lot about genomic data analysis that depends on matrix multiplication. So the objects that somebody might be using in genomics is not necessarily the best way to format that data and store that data is not necessarily a tidy data frame. However, what happens is usually after you do some kind of genomic data analysis, such as dimensionality reduction, you get to a point where you can store your data in a tidy data frame, and then it's ideal to essentially leverage the Tidyverse framework for data exploration, which is what it was built for.
Okay, great. Thank you. Something on a different tone. Do you think the academia is the main environment to work in if you're a student or a professional who's willing to work on this kind of data? Oh, that's a pretty huge.
The main environment, as opposed to industry. Does that mean industry or government or a nonprofit? So I'm going to assume that the person is considering different job sectors, and they want to know if I want to do genomic data analysis, is academia the only place you can do this? No, by no means. For example, Genentech in the US, in California, has recently hired a lot of great Bioconductor developers to develop Bioconductor data infrastructure and packages and be able to support the company, but then also being able to give back to the community. So it is true that I am in academia, but by no means I think do you have to be. I think there are a lot of opportunities, both inside and outside of academia, to be able to work in this with this kind of data.
Yeah, actually, I do think that these two things shouldn't be completely separated. Actually, I mean, it's a nurture thing between the two. And we had also a little discussion about business and opportunities, freelancing, and life science academia. And I think the two things are intermingled. And thank you for this take of yours. Okay, so let's see if here I can slide down. Okay, this is down. Well, this is pretty technical. What is the advantage of k-means over KNN, SNN graph clustering for single-cell RNA sequencing data?
Yeah, that's going to get pretty technical.
Yeah, we can skip to the next one.
Okay.
In your opinion, is single-cell RNA-seq delivering on its promises, or a bit of a hype that will be replaced by less problematic technological developments?
Oh, these are great questions.
Yeah, this
wow.
is a great question, actually.
Okay, so my opinion is that not everything that we have been doing with bulk RNA sequencing or microarrays needs single cell. So there are a few questions, quite a few questions that are ideal for single cell. And there are some questions, biological questions, that bulk RNA sequencing is more than okay and more than sufficient. And so I think in some ways, single cell is delivering on its promises because it is introducing us to be able to see data on a level that we've never been able to see before. And I'm really excited about new technologies as well, like spatial transcriptomics that allow us to kind of see another dimension as well. But it is true that single cell is, in the large scale of genomics, it's one type of technology. And yeah, it will probably change over time. Less problematic technological development soon. So I think the answer to that is with every new technology that's developed, there will always be problems. There will always be biases. There will always be technical artifacts in the data, no matter how great the technology is, in my opinion. The biases that we saw in microarray are still to a certain extent exist in bulk RNA sequencing, and those still to this day exist in single cell. There are different types of biases, and we've gotten really good at figuring out how to correct for a lot of them, but you should always expect problems every time a new technology comes out.
Yeah, especially pushing the limit, probably it's what we need to advance, and right. Pushing the limit means working on the edge. So right. That's why there are problems. Yeah. We have time probably for another couple of questions.
Sure.
Well, this is probably a Python user would argue, sorry to say Python in this conference, but that the Seurat is the de facto standard package for single cell. I mean, there's compile. But as a bioconductor advocate, how do you feel about Seurat now being the de facto standard package for a cRNA sequence analysis? Can the OSCA pipeline catch up in stage, in usage?
Well, I'm going to answer this question by saying I think the Seurat pipeline is wonderful. I mean, I think a lot of people use it in the way that it should be used, and I think it has a lot to offer. And I don't mean to suggest, or I'm not trying to take away users from the Seurat pipeline. I just, I want to be an advocate for Bioconductor to kind of showcase what Bioconductor has to offer. So when you look at Seurat, it's essentially, one big package, and it's not 1,900 packages. It's not, in the world of single cell, there are at least over 100 single cell packages on Bioconductor. And so it's easy to kind of get lost in how all of these packages work together, as opposed to Seurat, which is essentially one package. Now, there are benefits and disadvantages and advantages of both. So in Seurat, it's kind of like, it's harder. If I want to change out the normalization method, if I want to change out a different portion of that pipeline, it's not very easy to do so. You kind of just have to work within the Seurat framework, as opposed to Bioconductor offers a lot more diversity in terms of the types of methods that you can use. And so if you're somebody who wants to kind of explore the data with different types of methods, for example, maybe the normalization that you're using for your data violates some kind of assumption, and you know that in your data, but you want to switch it out. With the Seurat pipeline, it's a little bit harder, but with the Bioconductor pipeline, everything is modular. Everything is allows you to interoperate very easily. Output from one package is easily input from another package, and you can easily switch things out. And so I think it depends on the type of experience that you're looking for. To answer the question about catching up, I mean, I think Bioconductor kind of speaks for itself. I mean, if a user wants to use it, I think that's great. I think it has a lot to offer personally, and this is the framework that I choose to operate in. But similar to the discussion about whether you use R or Python, I mean, you should use whatever you need to be able to extract information from noisy, messy data. And if that's Seurat, that's fine. If that's Python, that's fine. I think Bioconductor has a lot to offer, though. So that was my goal with the talk.
Okay, I see that you decided Python here, and also we had this new question actually, but you already mentioned it's Python, and it's good though. But I would focus your attention on, for instance, this question, the last question. What are the key factors to develop? Oh no, sorry, I had in mind this one about education, and how easy do you feel it is to start with Bioconductor if one is not a technical person? I know that you're involved in a number of education and technical and learning experiences. So probably I wanted to hear your take on this.
Sure. So I really started to use Bioconductor during my postdoc in 2013, and I felt like it wasn't really easy to learn at the time. However, I think there's been a big recognition within the Bioconductor community that that's a big problem, and people are working very hard to change it. So for example, with my own contribution of OSCA, the goal was to be able to make it as straightforward as possible for a user who is not technical to be able to analyze or go from raw sequencing reads to identifying what the cell types are in his or her data, if you're willing to, read this really cool book. And so I think it's becoming easier, and I think there's a really big recognition that this is important for being able to allow new users to come into the community and leverage the power of the software.
Okay, thank you, Stephanie, for your talk, and it was very, very nice. And thank you for also referencing our city at the beginning of the talk. Milan has seen the Duomo, that was great, and unusually empty. So that's really weird.
I know.
Okay, I invite everyone to make a good talk. That's great. Thank you. Okay, now we have basically a virtual break. We can meet the sponsor, networking together with all